Thank you, Nick. That was really enjoyable. And you brought up Al taking lots of pictures, and putting this presentation together was a bit of a learning experience for me for two reasons. One, I'm not very good at taking pictures of the people that I'm with when I do field work, and I'm always then scrambling to try and find them. And also technology changes, and I know that I have pictures of work that I did with Al on CD-ROM, archaic file forms, and I can't even read a disc anymore. And so anyway, it's I guess what was a lesson to me about taking more pictures and trying to stay up to date on how to access them. And Al was always somebody who, like you said, loved to take pictures. I have lots of pictures of my crew and the places that we did sampling and the fishes that we were sampling that Al took. Uh, and some of the pictures that I have in my presentation are thanks to Al. So I'm going to try and share my screen. So what I wanted to talk about, well, first of all, thank you for letting me give a presentation talking about Al's work and also how Al's work impacted my career and the work that I've been doing. And so first of all, so the title of my presentation is Repeat Sampling Occupancy Models in Detecting Rare Fishes. And hopefully through this presentation, I'll be able to show why surveys for rare fishes are important, how we go about doing those, and where Al was very instrumental in shaping the work that I got involved in and others got involved in, and how we thought about this problem. And Nick gave a little bit of a preview into how Al's PhD work really led us on a certain path forward to do better work, to design better field surveys, and to come up with better results, and ultimately science advice to those people who are responsible for managing biodiversity and species at risk. So are you able to see my second slide now? Or am I still?
Not yet.
I'm sorry. I'm having some issues moving through my slide deck here. Sorry. For some reason, I get frozen out of presentations after a certain period of time, and so it's giving a bit of issues. So we're still able to see my first slide?
Yes. Yeah.
Okay. So I'm going to go forward without you seeing my camera, and hopefully we can do this.
This might be has two screens on.
Do you have two screens going by any? Oh, there it is. Okay, we can see them now.
Great. So why are surveys for rare fishes or imperiled species important? I thought I'd just sort of start there, and that gives you an understanding about why we care about this, beyond the fact that they're really cool habitats to sample, and they're really cool fishes. So this is really essential information for when we get to species status assessments and listing decisions. So this speaks to Al's involvement on the freshwater fishes subcommittee of CSIWIC, which he encouraged me to join. And then also CSIRO, which does the same role in Ontario, in which Al was a longtime member of. The picture at the beginning of Nick's slide brought back a memory of Al telling me that he saw a draft by Ed Cott of a draft CSIWIC status assessment for greater red horse. Unfortunately, that status assessment report was never finished, and we never got around to assessing the status of greater red horse, which seems to be relatively secure in Ontario, but I know in other neighboring jurisdictions it isn't. So this information is important for identifying those areas that we need to, those habitats and areas that we need to protect to conserve fish biodiversity. When I think about SARA or ESA permitting, this information, this site level information, is it's integral and important for impact assessment. And then hopefully with species at risk, where our intent is recovery planning and getting recovery actions done and recovering these species. And this sort of information is essential to inform and direct those actions, and then ultimately to follow up and see if we did a good job. So on my third slide here, starting to think about imperfect detection and the design of rare fish species. And I wanted to start, linking back to an early part of my career where Al was strongly involved, and whether that was encouraging me and taking me to the Red Site Days recovery team meetings to be a member when I worked for him as a species at risk biologist, or earlier when I was doing work with an undergraduate student, Jason Lean, studying channel darters. Al was coming out in the field, helping us to give direction and insight on how to sample the species and just bring a general high enthusiasm to the work. And then the other cover page I have is, my first real effort to go out with Al and do a survey. And so he took me out shortly after I started working for him as a species at risk biologist. We got in one of the M&R sedans, grabbed a green seine net, and headed out to Eastern Ontario looking for the eye picker or the cutlet minnow. And it was a wonderful couple of days with Al, looking for the eye picker, looking for channel darters, and going back to all these historical collection record areas and seeing what was the status of cutlet minnow. But hearkening, I guess, back to how we used to do work, we didn't think too much about how robust our sampling was, and getting at what Nick was talking about in regards to, we're species there, but we just weren't looking hard enough. And a lot of what we did was simply a single pass survey design. And it was sort of how people did work, more than 15 years ago or 20 years ago. And it was, just a typical design. It was cost effective. We spent a lot of time driving all over the landscape, going to these places, and spending a lot of time at an individual site sort of gets in the way of getting more samples or sites done. But as Al educated us, species detection is imperfect. This could be because your gear is only so effective at collecting a certain species. For fishes at risk, low abundance is a problem for just in terms of the probability of encountering them. Some of the species that we think are cool or interesting, they're cryptic. They have the habitats they use. They could be benthically orientated, so they're hidden around rocks. So they're hard to find. Or maybe they have a diel behavior pattern, and they're really only active at nighttime. When we tend not to go out and collect fishes, we prefer to be, reminiscing and having a beer and dinner and all that sort of stuff. But what are the consequences of not thinking about species detection being imperfect? Well, we have incomplete species lists when we go out to survey areas. Our species richness estimates for watersheds or wetlands might be biased. And then when it comes to understanding the relationship between species and their habitats and their environments, we end up with biased or flawed species distribution models. So how did we used to think about this problem of how many samples do I have to collect to be confident that a fish is at risk is present in the area of interest? The initial method that we used was randomization. So go out to an area, collect a pretty decent sized number of samples, and then take that data set and resample them over and over and over again so we can develop these probability curves, these relationships between effort on the x-axis and probability of detection along the Y. Mark Poos, now Mark Posch, did this for his work in the Sydenham River with black-struck top minnow, spotted gar, and eastern sand darter. And I used the same approach to come up with a backpack electrofishing protocol to detect American eel in eastern Lake Ontario watersheds. And so you say, I want to be 95% confident in being able to detect a species. And you go across and you look up from your x-axis and you get an estimate for how much sampling do I have to do to be 95% confident that we're going to detect a species if it is there. Well, Al's work in his PhD and then later with the postdoc that he did with, or visiting, maybe it was a visiting fellowship at the government lab, they still call them that, to provided us a path forward to do a better job of coming up with these sort of estimates of how much sampling effort do we have to put in to confidently detect fishes at risk, but also a framework so we can better understand those important factors which are going to influence how well gear perform in surveys. So to give you a quick overview of this approach and how we follow it. So first of all, we go to sites and we collect replicate samples. These can be temporally replicated, so going back multiple days in a row. They can be spatially replicated within a habitat unit, or it could be something alternate, like multiple observers all sampling at the same time, or multiple gear being deployed concurrently. We also collect information on those variables that we hypothesize are important. So this is not data mining. This is not going out and collecting the kitchen sink, but this is being thoughtful beforehand and coming up with good hypotheses about what we think are important in terms of determining where species are, and also those variables that might affect our ability to detect a species when we go sample. So then we take this data and we jointly model detection probability and site occupancy at the same time, and therefore gives us that opportunity to adjust our occupancy models based on the likelihood of us detecting the species if it was present but not detected. We can then use the parameter outputs from these models as inputs into other equations, so we can estimate the minimum number of samples or surveys that we have to do to be confident at 95 or 99% confident. And we can also look at survey designs in the context of, if we want to use this as part of a long-term monitoring program, how confident are we that we're going to be able to detect changes of certain magnitudes? And often we end up defaulting to the IUCN criteria of 30, 50, or 70% declines. So that's the framework that Al showed us forward. Al's research on this was pioneering and it was inspiring. And what I tried to do with this figure, and this is just a subset of those publications that I know of projects that either I was involved in, other grad students, or other researchers that really were inspired to study the world differently and study Ontario fishes and their monitoring and their habitat associations, their interactions with other species within the context of this occupancy framework. And that really is Al's legacy right there, from my perspective. So the next thing I wanted to do in this talk was just to go over a couple of projects that Al and I did together, and to sort of maybe give a little bit more detail on some of the results of research that he was involved in, that as a research partner with me. So the first one I wanted to talk about was eastern sand darter, and I thought this was appropriate because Al did so much work on eastern sand darter and really pushed our knowledge and management of eastern sand darters forward. And so the one project that we did came out of an accidental discovery of eastern sand darter in Lake Ontario. Previously, we believed that eastern sand darter was only found in Lake Erie, southwestern Ontario, and then off in Quebec. And while my staff was out looking for pug-nosed shiner, they ended up finding eastern sand darters. Don't ask me why they were sampling habitats that got them eastern sand darters when they were looking for pug-nosed shiners, but luckily they tripped over them. And this led Al and I to lead another project in collaboration with DFO to go and more intensively look at the population that was discovered in West Lake in Prince Edward County. And so this led to Al and I writing up a note describing this discovery and checklist, and then going forward on a project where we looked at two different seine methods that were being used to survey sandy habitats for darters at risk in Ontario. And that was the regular bag seine and then the beach seine. There's I think a really good picture in the top right that shows, probably what eastern sand darters look like naturally. And that's, like the one that Nick showed with them half submerged in the seine. So in terms of, how did we design this work? How did we do this work? So we used a removal design. So again, a replicated sampling design, 5 repeat hauls over a same transect to provide us with our detection history information, which was essential for occupancy modeling. We used a bag seine along 10 meters in length, which was it's a typical gear for river surveys, and this is about a typical amount of habitat that's sampled for rivers from eastern sand darter. And then we also used the beach seine, which we had been using for channel darter surveys in Lake Erie. We did 69 sites adjacent to the southwest end of West Lake, which is in Sandbanks Provincial Park. And these are all habitats that run along a large barrier sandbar. And this is where we find eastern sand darters in this part of the world. And the graph on the bottom right sort of shows you that the detections that we get by doing this removal sampling, one, that if we had just done one lesson is that if we had just done one survey, there were likely a bunch of sites where we wouldn't have detected eastern sand darter with either gear, and also that this pattern of detection is variable over a number of replicate sites, and that there were differences between the two different gear that we used. So now I'm just going to get into the stuff that Al really shone on, and that was using these occupancy models to provide useful results. And so what I've presented here on the left are two tables which give estimates of detection probability and occupancy from the two different gear types of the same habitats. So the main thing that we learned is that for sampling these sort of sandy habitats, that we had a greater detection probability, likelihood of collecting eastern sand darter using the beach seine. And this was significantly different. And so that was an important learning for designing surveys going forward. The other thing that we were able to use from this result was a follow-up work that myself and Tim Haxton led in Westlake and nearby Wellers Bay, was to see if environmental DNA was a good sampling method for detecting eastern sand darter. And so we used a replicated design over the same area a couple of years later to look for eastern sand darters. And by using replicated data, we were able to estimate for all 3 gear types or sampling methods, what's the likelihood of detecting eastern sand darter. And while certainly we learned that we could use environmental DNA to detect eastern sand darter, that staining gave us a greater likelihood of detecting the species. And you can see that in the right graph. So much of the work that I did with Al was using a seine. Low tech. Al was, I think, while we all recognized the value of technological advancements, Al also liked the low tech, old school way of doing work. And he always used to joke that so much of the work that we did was basically using rulers and bits of string. So the second example that I want to talk about is the last project that Al and I worked on, and he and I were in progress for developing a manuscript um, to apply occupancy modeling to look at how good of a job pelagic trawls do at detecting wetland fishes. And the data set that we had available to us was a data set that Anita LeBaron and I collected along the Trent River. And so the pelagic trawl has a benefit or advantages over the seine for those habitats that are either too deep to seine or the bottom is too soft to easily drag a seine through. And so you can deploy this pelagic trawl and drag it through wetland habitats. And if you follow the way that Al advises you work, you take replicate samples. And so our data set, we had 66 sites along the Trent River, 50-meter transects, and 3 repeat hauls. So this slide just gives you an idea of the types of habitats that we would sample. Often they are offshore. Sometimes these are flooded islands that would otherwise not be accessible, or they'd be heavily vegetated and very difficult to drag a seine. And so here's just a few pictures of me and Anita and I deploying the seine to give you a sense of how this works and dragging it, pulling it through. So getting to the results of that study, we did detect, I believe, about at least 22 species, variety of species, including black line shiners or black nose shiners, or black chin shiners and pug nose shiners, as well as some basses and perches and sunfishes. We also found that species detection was imperfect. So if you look at this species accumulation curve, you could see that pooled samples versus a single, so pooled 3 samples versus a single sample, allows you to more rapidly and fully detect the species that are present within these habitats. And looking at some mean differences, that as you got to you add one additional pass, you detected on average more than 1 species at a site, and then if you added a third, not quite enough additional species. So this really highlighted the need to be cognizant of imperfect species detection and to apply occupancy models to come up with guidance for designing, monitoring, and sampling strategies moving forward with the pelagic or mountain trawl. So here's some summary of some results that Al worked up. Our occupancy models, before we got into the business of parameterizing our models, we talked a lot. We spent a few meetings talking about what we wanted to accomplish with this modeling exercise, what species were suitable for us to use for this modeling approach, and those habitat covariates that we felt were essential to be included in occupancy models. And this is really a great part of collaborating with Al, was that he loved to talk about why we're doing things, what we're hoping to achieve, and what were really the best ways of doing things and thinking about problems. So we talked a lot about what parameters we wanted to include, how suitable certain parameters were. Did we have to transform them? Did we have to create composite parameters so that we did a good job developing models? And so this table just provides you with some output from that modeling exercise. Again, it's the story. Again, species detection is imperfect. That variation is different among species. It reflects abundance. It reflects habitat associations of species, and it reflects the differences in the vulnerability of species to our gear. In some terms of what are some of the best models? Our null model, which is that none of our hypothesized covariates really affected species detection probability too much, but we did see, and not surprisingly, that some of the macrophyte and substrate covariates did influence the occupancy patterns of the species we got into. And then we use this data to kind of explore and address the question of, if we use this gear and we use this sampling strategy, what is the power that we're going to be able to detect changes in abundance over time? So we took those parameter estimates of occupancy, detection probability for the different species that we modeled, and we used a power analysis to see about what was the likelihood that we're going to detect changes of 30%, 50%, or 70% in the distribution of these species between two sampling periods. And my little heat map here table on the right just sort of gives you an overview of those estimates. And for some species, we have a lot of confidence. So like black chin shiner, blunt-nosed minnow, we have a lot of confidence that we'll be able to detect further declines or increases in the distribution of species. Whereas something like the pug-nosed shiner, our sampling design we're much less confident, whether that's an insufficient number of passes or that the species just isn't found in enough sites. And so that's sort of a preview to a paper I'll be working to finish this year of work that Al and I did together. So my last slide here is my parting thoughts. And this kind of gets back to one of the things that I thought was great about working with Al. Al was a deep thinker. And he loved to find insights from other deep thinkers, you know, and he would as a mentor and a collaborator, he did a wonderful job directing me to guidance, knowledge, pathways forward on how to do things. Whether it was Roger Green's early book on sampling designs and methods for environmental biologists, his little checklist of things you should do before you do work, David Anderson's primer, ideas about how you construct models, confront models. I don't have a Ecological Detective was another great book that Al passed on to me, and of course, you know, directing me towards Darrell Mackenzie's tomb occupancy estimation modeling. And so, you know, one of the things that Al used to like to bring up when people were having problems with putting their models together or writing our code or using presence, he always used to laugh about one of his favorite lines of advice in phi.org, which is a website for doing occupancy modeling and providing insight. And it was RTFM, which was read the bleeping man. And I liked that, and I thought it was a good example because Al always brought humor and levity to work, and that was always appreciated. So that's the end of my project or my talk today. Thank you.